the ongoing lecture series on plant tissue culture, I have discussed about the history of plant tissue culture, that how it develops into a powerful biotechnological tool over the period of time. Then I discussed about the characters of an ideal plant tissue culture laboratory, the steps that are involved in setting up a plant tissue culture. Then we discuss about the callus and suspension cultures, how is the growth rate determined, what are the various techniques by which you can determine the viability of the cells in the cultures, and applications of these cultures. After that, I started with protoplast culture, wherein we discussed about various techniques that are involved in isolation of protoplast, then how the protoplast are fused, what are the factors that affect the protoplast culture, and applications of protoplast culture. So friends, in continuation with the lecture series, today I am going to start with another important technique under plant tissue culture, that is hairy root culture. Now, as the name indicates, hairy root, it means this is a culture of the roots which have hairy characters. Now, in this lecture, we are going to discuss in brief about the historical background of hairy root culture, then what exactly are hairy roots and how they are produced, then what is the procedure to perform a hairy root culture or how these hairy root cultures are maintained, and eventually we will discuss about the applications of hairy root culture. Though the term hairy root culture or the hairy root was originated in late 20th century, but they appear on the scientific chart during 1930s and 60s, when it was found that there are certain fruit crops that are infected or they exhibited their roots with a huge development, huge multiplication, huge branching of into the lateral roots, and they showed the appearance of a network of roots. So this was the first instance in 1930 when such kind of growth of the roots was observed. Riker and his group in 1930 for the first time identified the causative agent of this particular hairy root appearance, and they identified that the causing organism was Phytomonas rhizogenes, which was renamed by that group to Agrobacterium rhizogenes, and as per the latest nomenclature, this is now known as Rhizobium rhizogenes. So we can say it started in 1930. Then Chilton in 1982 for the first time showed that the tDNA or tumor induced DNA or transfer DNA of RI plasmid of that particular bacteria, Rhizobium rhizogenes, is mainly responsible for the occurrence or the production of the hairy root by a particular plant. 1990s was the era wherein the understanding of the mechanism, that how this RI plasmid causes hairy root to occur in a plant and what is the role of different genes that are present in this particular RI plasmid are responsible for the production of hairy root in a particular plant. Now we move on to what are exactly hairy roots. Now these hairy roots are also known as crazy roots, because as I told you that during 1930 it was found that certain fruit crops, they develop hairy roots, and those roots have high lateral branching and they form a network like arrangement, and the growth was very fast, and therefore we call them as crazy roots. And these roots have a very high growth rate in comparison to the normal roots, and as we discussed, they have a high lateral branching which causes high profusion of these hairy roots, and this high profusion and high rate of lateral branching gave them a hairy appearance, and that's why the name hairy roots is given. And another important character of hairy roots is that these roots lack geotropism. we know that majority of the normal roots are geotropic, but these hairy roots are, they don't follow geotropism, they lack geotropism, and they can grow in any direction basically. Now, how they are produced? So, as it was discovered by Riker, and that the causative agent is Rhizobium rhizogenes, and because of the scientific work that was done in 1990s, we now say that the transfer DNA, that is the fragment of RI plasmid, Rhizobium genus plasmid of that particular bacteria, is incorporated into the plant genome, and the genes that are present on that tDNA, they lead to the production or induction of the hairy roots in the plants. So, basically it is nothing but it is a sort of infection to the plant that is caused by the Rhizobium rhizogene bacteria. Whenever a plant is being injured, it releases certain important phenolic compounds which attract these bacteria, and at that wounded side they get incorporated into the plant cell, and they release their tDNA, which eventually causes the production of that infection, leading to the formation of hairy roots. Now, it has been found that there are different strains of Rhizobium rhizogenes, and these strains have been categorized on the basis of the opine production. Now, opines are nothing but these are low molecular weight compounds that are produced by these bacteria, and these opine compounds are responsible for providing energy. They act as carbon or nitrogen source for the bacteria. So, depending upon the type of the opine production, there are different strains of Rhizobium rhizogenes, and these are octopine, menopine, cucumopine, agropine and nopaline. Now, amongst all these strains, agropine is the most favored one, or I will say it is a strain of choice for inducing hairy roots in a plant because of its high ability to induce the hairy roots in a particular plant in comparison to the other strains of the bacteria. Now, as we discussed that there is an RI plasmid in the bacteria, and that is responsible for the induction because this RI plasmid contains tDNA. So, as you can see in the structure, this tDNA is having at the corners borders which demarcate from rest of the areas of the RI plasmid. Now, these borders are nothing but these borders are 25 base pair repeats, and these borders, as I told you, that they demarcate tDNA from rest of the plasmid. Now, if you see into the structure of RI plasmid, this tDNA has two flanks or zones, we can say. One is left flank, which we call as TLDNA, and second is TRDNA. And in the center is a small fragment of tDNA. Now, this small fragment of tDNA, or we can say, is replaced by the desirable gene which we want to introduce into the plant genome. So, as we can see that the TLDNA or the left flank of RI plasmid or tDNA contains certain set of genes which we call them as role genes, and these are nothing but role A gene, B, C and D genes. Now, these genes are responsible for the induction of hairy roots, and amongst these genes, role B is the most important one. Whereas on the right flank of tDNA, we can see there are certain set of genes which augment the production of auxins once the plant is infected by this RI plasmid. Apart from this tDNA, the other zones in plasmid is a virulence gene zone, or we call them as VIR genes. Now, these genes are responsible, or I will say they help in transferring of the tDNA into the plant cell. And then we also have opine catabolism genes which are responsible for the catabolism of opines, that is opine production. There are certain genes which are responsible for the metabolism of opines and so on and so forth. So, by and large, we can say that RI plasmid is broadly categorized into tDNA region, VIR genes and the opine catabolism or metabolism gene areas. Now, the question is how this tDNA is transferred into the plant cell. Now, as I discussed that as soon as the plant is injured, it releases certain phenolic compounds which attract this Rhizobium rhizogene bacteria. Now, once it get attracted towards that those phenolic compounds when the plant releases them after injury, CHVB genes of the bacteria, they get activated and they start producing a protein that forms the cyclic beta 1,2 glucan compound. Now, this compound is released or produced in the periplasm, that is the space between the plasma membrane and the cell wall, and this compound basically acts as the anchor site for the attachment of the bacteria. So, bacteria basically attach to this beta 1,2 glucan molecule that is produced by CHVB gene of the bacteria. Now, once the bacteria gets attached to the plant cell, the virulence genes or the vir genes of the bacteria, they get activated and they cut the tDNA in the in that particular RI plasmid of the bacteria. Once the tDNA is cut, then vir D1 and D2, they basically they forms a vir D2 tDNA complex. Now, once this complex is formed, other vir proteins like vir E2 and vir F, they also join to that complex in order to protect that complex from the enzymatic degradation. Now, once this complex of vir D2 tDNA, vir E2 and vir F is formed, then vir B proteins get activated along with vir D4, and both these proteins, they together form a type 4 protein which we call as T4SS. Now, this protein is nothing but it forms a channel between the bacterial cell and the plant cell, and or we can say it forms a pore between the bacterial cell and the plant cell that allows the complex of tDNA and other vir proteins to pass from the bacteria into the plant cell. Now, once this complex of tDNA with D2 and E2 proteins, it enters into the plant cell, they again form complex with VIP1 proteins in the plant cell, and this huge complex we call them as super T complex. Now, super T complex, or we will say VIP1 proteins, they are responsible for guiding this tDNA with D2 vir E2 complex to the nucleus of the plant cell. So, super T complex then get transported to the nucleus of the plant cell. Once it is inside the nucleus, vir F protein which are also a part of that tDNA complex, they get activated and they destabilize the VIP1 proteins. Once this get destabilized, the interaction become very weak and this protein dissociates from the complex. Further, vir F protein also destabilize the vir E2 and vir D2 interactions with tDNA, thereby totally uncoat the tDNA from the vir proteins, or simply we can say vir F proteins help in removing all the other proteins that form complex with tDNA, thereby releasing tDNA into the nucleus of the plant cell, and then this tDNA get incorporated into plant genome and produce its effect. So, here is a diagrammatic representation of what we have discussed till now. So, here you can see that once the tDNA is released from RI plasmid, it get interacted with vir D proteins and vir E proteins. Then vir B proteins along with vir D4, they form a T4SS protein channel through which this DNA complex get entered into the plant cell, where it interacts with VIP1 protein which guide them towards the nucleus. Once they enter into the nucleus, vir F proteins, they act upon this super T complex and destabilize it, releasing the tDNA from all the vir protein and VIP proteins, and eventually that tDNA get incorporated into the plant genome. So, this is how the infection or the tDNA of RI plasmid gets incorporated into the plant genome. Now, what are the advantages of hairy root culture or hairy roots if we talk about? As we discussed, they have a rapid growth rate in comparison to the normal roots. So, if we say that you want to grow a root part of a particular plant, or we say that the active constituents are produced by the roots, we can induce hairy root cultures in those roots to enhance the production of that particular compound. Now, these roots provide a stable production because they are grown under controlled conditions, so they have a specific stability in terms of genetics, in terms of biochemistry of the compounds that are produced. Then, as we discussed, these hairy roots help in enhancing the production of secondary metabolites. They are grown in controlled conditions, so the temperature conditions, the pH conditions of the culture are normally maintained at a stable value. Therefore, we can say that they grow in a controlled conditions, or the stability of these hairy roots are there. Then they are independent from phytohormone. So their growth does not depend largely upon the content of the phytohormone that is present in the media, because they are largely dependent upon the tDNA of the bacteria. These hairy roots are versatile. When we say versatile, then they not only enhance the production of secondary metabolites, but lot of other type of compounds or biological agents can be produced by using the hairy root cultures, which we are going to discuss in detail when we will discuss about the applications of hairy root culture. Then once these proteins are produced by hairy root cultures, they act as a low cost technique for the purification of such proteins. When we talk about versatility, in addition to secondary metabolites, they also help in producing certain type of proteins. And we know that the techniques that are used to purify those proteins are very costly, but hairy root cultures act as a low cost technique for the purification of those proteins. Now, despite all the advantages of these hairy root or hairy root cultures, but when we talk about the commercial applicability, there are certain problems when we talk about the scaling up of the hairy roots for the commercial production. Now, hairy roots have a very intricate morphology, and when we talk about intricate morphology, we discuss that they have a high lateral branching rate when these are growing. So, because of that high lateral branching, these roots form a very dense network of root hairs, and because of this dense network of root hairs, the roots sometimes they form aggregates, and these aggregates basically prevents the mass transfer, they prevents the transfer of nutrients to the growing cells which are present around in the center of those aggregates. Further, they also prevent the exchange of oxygen, because oxygen is something that is required for those growing hairy root cells. So once the dense network is formed, there is a resistance, there is a hindrance in the mass transfer, nutrient transfer, as well as the oxygen transfer rate. Then, since there is a hindrance or limitation in nutrient transmission, in oxygen exchange, there is a non-uniform growth in those aggregates, because oxygen is not properly reaching to the central core of those aggregates, which develop certain senescent spots. Senescent spots are nothing but these are the patch of the cells which are getting dead. Therefore, they exhibit a non-uniform growth when we want to scale up the growth of hairy roots for the commercial applicability. Now, these hairy roots are highly sensitive towards change of any kind of temperature or change in chemical composition of media or any kind of shear stress into the culture vessel. Now, because these roots have very intricate morphology, sometimes there is a variation in the temperatures in the different zones of those root aggregates, which again causes the hindered growth of the roots, or causes the limitations, or causes the prevention of the production of secondary metabolites. Then chemical composition again changes with the as the media get exhausted. So if it is not replaced periodically, then again it will have a negative impact on the overall growth of the hairy roots and the production of the secondary metabolites. Now, since these have intricate morphology, and they resist the mass or nutrient transfer or oxygen exchange, we have to provide some sort of shaking of that culture so that there is a proper circulation of nutrients, circulation of oxygen. Now this kind of circulation is done with the help of impellers, and these impellers sometimes provide a shear stress onto the roots, which again have a negative impact on their growth as well as production of the secondary metabolites. Now when these hairy roots, they grow in a commercial vessel which we call as bioreactors, then their increase in volume and pressure in the scale up, that also adds up to the overall pressure onto the roots. Now when this kind of pressure on the roots is increased, their overall ability to produce the secondary metabolites, their overall ability to produce the proteins which we want them to produce goes down, and the overall cost to product ratio that changes, and the whole process become costly. So these are some of the problems while scaling up the hairy roots. Now various bioreactors have been designed to overcome such problems, and the most common, or the I will say traditionally using bioreactors, are stirred tank bioreactors, bubble column bioreactors, airlift bioreactors, hybrid bioreactors, and some of the upcoming or the latest bioreactors include nutrient mist or trickle bed or mist bioreactors, we call them. Hydraulic driven bioreactors are there, immersion bioreactors are there, slug bubble bioreactors or micro bioreactors. Now some of these bioreactors have been discussed in one of my previous lectures on suspension cultures, but we will discuss all these bioreactors in detail in a separate lecture that will be dedicated only to the bioreactors used in plant tissue culture. Now we move on to the factors that affect the growth in these bioreactors, and these are aggregation and adhesion of the roots. Aggregation because they have a very intricate morphology, high lateral branching, which leads to the formation of aggregates. And some of the cells, they adhere to each other because the cells have natural tendency to adhere, and therefore such kind of adhesion leads to aggregation. Sometimes cells also adhere to the walls of the bioreactor, which pose another kind of problem. Then mixing of the culture media for proper gaseous exchange, for proper nutrient absorption by the cells, and viscosity of the media also plays an important role in overall growth of the culture. Then aeration and shear effects, and the type of impellers we are using in the bioreactors, that also affect the overall growth of those hairy roots. Now we will discuss all these factors in detail. So coming on to aggregation and adhesion. Now cells have a tendency to produce certain polysaccharides, and these polysaccharides are responsible for cell to cell adhesion, and one of such polysaccharide is pectin. Now these polysaccharides not only are responsible for cell to cell adhesion, but sometimes they also act as a source of cell to cell communication, which eventually causes the production of secondary metabolites. So therefore we can say that these aggregates or aggregates are also important, but up to a particular threshold value. So as you can see that large aggregates, if their size range from 2 to 10 millimeters, then they cause the problem. Problems like there will be less oxygen supply to the innermost cell, which gradually loses their viability and gets converted into senescent patches in those particular aggregates. Further, in order to have a proper oxygen supply, proper nutrient supply, we sometimes expose those medias to a high speed mixing using impellers or high speed bubbles in bubble reactors. Now this kind of high speed mixing or bubbling through the media in the bioreactors also causes certain cells to adhere to the walls, and slowly they start adhering to each other, forming aggregates at the walls of the bioreactors. Now once these aggregates of the wall, are formed on the walls of the bioreactors, they again inhibit or limit the oxygen transfer, they limit the nutrient transfer, and they eventually change the overall viscosity of the media. But as we discuss, these aggregates are important for the proper growth if they are below the threshold value of the size, because as I told you that these small aggregates or single cell, basically they help in production of secondary metabolite through cell to cell communication. Now if these aggregates are too small or the cells are present in single, then sometimes they lose their viability and there is less production of secondary metabolites. So friends, in the next lecture we are going to discuss the remaining factors that will affect the growth of hairy root cultures in bioreactors. Thank you.